Okay, so I think the issue is sorted now. I think the screen is visible. Is it visible? And am I audible clearly now? Okay, okay. So really sorry for the technical glitch. I don't know what happened. So I think we'll get started now. So yeah, as mentioned, we'll have one more revision session tomorrow. Whoever will be able to join, I'd be happy to have you in the last session tomorrow also. So I think you received a announcement today by mail that there will be a session tomorrow also. So that will be the revision session, and today we will just focus on this week 12 content. So okay, so as I was discussing, I was asking you what was bioinformatics. I'm sorry, I just saw one response which told obtaining information from biological samples. I don't know if there was some other responses because I was switching from one device to another. So if there were more responses, you can please again respond in the chat box. I think that one response was from Shagupta Khan. It was telling that we are obtaining information from biological samples. That was bioinformatics. So with that little introduction, here we have what is bioinformatics. Bioinformatics is generally conceptualizing biology in terms of molecules and then applying informatic techniques. So informatic techniques generally uses math, computer science and statistics. So what we are basically going to do is we are going to get a quantitative analytical data so that we can get some understandable and reportable numbers basically. Uh, because all these molecules and stuff, we have imaging techniques, we have other techniques for qualitative identification. So in informatics what we do is we go for a quantitative measurement. This domain generally integrates biology, computer science, mathematics, statistics and information technology. As in as the progression is there in every field, we also have the intervention of AI/ML in this field also. So all starting from basic mathematics to all modern information and processing technologies, everything can be integrated to biology, and that field is known as bioinformatics. And also one more thing that I wanted to tell. Okay, let me just check the PPT once. Okay. So actually, I think the animation did not work here, so you got the display of the answer. But yeah, the main thing that I wanted to differentiate here was the difference between genomics and metagenomics. So genomics, with only one organism, if you are going to study the entire set of genes present in that organism, it's genomics. Whereas in metagenomics, it's about the entire population in a given sample. You study about the genes of all organisms that is present in a given sample. So that is known as metagenomics. So with that, I think we'll go for the first practice question for today. So this is the first question for today. Okay, very happy to see very quick responses. Um, which of the following is not a commonly used bioinformatics tool for understanding biological data? We have Megan, we have Blender, Kime, and Blast, and all of you are telling it's Blender. So let's first check out if any of you know what's Megan. Okay, I'll just give you a few minutes uh, to think and answer. You can answer about any of the four options. If you know about Megan, you can type about Megan. If you know about Blender, if you know about Kime, if you know about Blast, whichever thing you know, you can just type in the chat box and then we'll discuss. Okay, we have a few responses. Blender is a program for molecular modeling. ANC used for metagenome analysis, and BLAST used to know the similarity. KIME is a bioinformatics open source pipeline, and it provides quantitative insights into microbial ecology. Okay, all of you are right. And yes, the answer is Blender. So Megan stands for metagenome analyzer, and as you rightly mentioned, we can analyze large metagenomic data sets. KIME is a open source bioinformatics pipeline for microbiome analysis from raw DNA sequencing data. If we just input raw DNA sequence that we have obtained after our sequencing, we can do the microbiome analysis using KIME. So BLAST stands for Basic Local Alignment Search Tool. So as the name indicates, we go for alignment. So that is what I think you mentioned as no similarity. Uh, so by performing the alignment, we'll know what is the local similarity between the two sequences that we have input. Blender in that case is used for molecular modeling, yes, but it's not very confined to biology alone. It's a 3D modeling software. So we prepare illustrations and all for our works, right? So all these illustrations, if you want to prepare it in a 3D way, you can use this software called Blender. So it's not a bioinformatics software. It's 3D and animation software. You can use it for any purpose. All the answers were right. So here is the next question. Okay, I've received responses from three of you. So before discussing this, I'll also ask another question like what is sequencing depth? Okay, one of you was telling total sequences obtained from the analysis is the sequence depth. Single fragments, how many times? Okay, how many times we are sequencing a single fragment is known as sequence depth. Okay, any more answers? Okay, let me define sequencing depth. So, sequencing depth is nothing but the average number of times a given position in the genome is sequenced. So, almost to what Shagufta Khan has answered, it's the average number of times a particular position in the genome is sequenced. So what we are telling here is how does sequencing depth, that is the number of times that a particular position is being sequenced, how does it affect the accuracy of this sequencing? So the first option says accuracy increases with increase of sequence depth. Accuracy decreases with increase of sequence depth. There is no relationship between accuracy and sequence depth. And accuracy increases initially and eventually reaches a plateau. So here what happens is sequencing depth. So as with the number of times that we are going to repeat this process, because this process has its own probabilities. So because with the number of times that we are going to repeat this, we'll have a very much reliable data. So we can now conclude that accuracy does have a relationship, and also that relationship is going to be something that is of increasing in nature. Okay, so with that we rule out that option B and C. Okay, with the definition of sequencing depth. But when you think logically, what happens is you are given a set of sequence, and how many number of times you are going to sequence the same position. At one point what happens is you are very thorough that okay, this is what is going to be in that place. Even after knowing it so thoroughly, if you are going to again sequence the same position, then the sequence depth is going to increase, but there is no change in the result that you are going to obtain, right? So what happens is, um, the accuracy increases initially with the increase of sequence depth, but after some time what happens is, it is what it is, you are not going to get any varied results after that. So it eventually reaches a plateau. So here the answer for this question is not A but D. Actually it increases initially, uh, and then it reaches a plateau. So here I have given what is sequencing depth. With this you can correlate. And here is the next question. So you can answer this question and also give in the chat box what's a microarray or a multiplexed PCR. I have discussed about microarray and PCR in, I think, in week 5 or something around. In one of the previous sessions, I have discussed about this. If any of you are not aware, you can let me know in the chat box. Or if you know what these terms mean, you can give that also. Any answers? Okay, we have two responses. Anyone who knows what is a PCR, a multiplex PCR, or a microarray? Any of these terms? I'm sure you should have at least heard about PCR, if not for the other two. Okay, then let me just explain what it is. So we'll come back to the answer. We'll just see the what the correct answer is. It's C. I'll first explain things and then we'll come back about discussing. Okay. So this is DNA microarray. So in this, what we have is we are going to hybridize. We have a healthy cell as well as pathological cells. We isolate the RNA from these cells and we do this reverse transcription. Okay, and if you do, you will get a cDNA and you're going to tag it and hybridize it onto a microarray plate. So in this, you can differentiate which are the normal cells, which are the pathological cells, and which are common and which are not present. So all these things you can study from this thing. What happens is you just have a plate like this. In this plate, you just take cells, isolate their RNA, do reverse transcription, and then hybridize. So for hybridizing, you will have the complementary stand in this thing. And after that, when you're going to detect it, based on the tags that you have given for each of this, you will get the signals. So when you have only the normal cells, you can see which are the normal cells from the color signals, which are the pathological cells, and in which that thing is not at all present. So what this helps you to study is that the presence of that particular gene, and also you can study multiple samples at a time. This technique is microarray technique. I have explained it in week 5 lecture also, I guess. And this one is about PCR. So PCR is generally a gene amplification technique. So you can make multiple copies of DNA using PCR. So what you do is you add all of this, the sample, primers, nucleotides, the buffer, and the most important thing is a Taq polymerase. I guess you remember what a polymerase is. That will help in this polymerization of the DNA strand. Okay, so you are going to add all of it here. So in the first step, what happens is your sample will denature, and then the primers that you have added will anneal, and then with the help of polymerase, the polymerase with the help of this nucleotides will elongate the strands, and then finally you will get two new copies. From one DNA strand you have made a two copies of this DNA sample. So similarly it will multiply for multiple cycles and you will get certain amplifications. And so this is generally a PCR. So PCR stands for a polymerase chain reaction. um, this process uses polymerase and will continue, and finally you will get an amplified product, DNA product. Okay, this is generally what a PCR means. In a multiplex PCR, what happens is you can add multiple genes, multiple DNA samples, you can add the corresponding primers to it, all of it will be amplified. So as the name suggests, it's multiplex. So you can add genes from, for example, strain A, strain B, strain C, and strain D. You add the corresponding primers of the genes that you need to amplify, and in the same tube itself, all of this will be amplified, which you, when you run on electrophoresis gel, you can see that you have obtained all four of them at the end of amplification. So this is a multiplex PCR technique. Okay, so here what the question is, what is the advantage of using microarray or multiplexed PCR over traditional PCR methods for detecting microbial communities? The options are microarrays and multiplex PCR are more sensitive than PCR based methods. They are speedier than PCR based methods. They can simultaneously detect higher number of microbial taxa, and they are more economical than PCR based methods. So regarding the economy, there's not going to be much of a difference because you are just again going to use uh, the same instrumentation, the same chemistry or the chemicals facility that is required. So that's not going to be a much of difference. If it is going to be speedier, yeah, it's kind of a speedier because here you do simultaneous analysis, but in other case what happens is you have to run one individual PCR for one individual sample, so it's going to be expensive in that way, or it's going to be time taking or easier in that sense. And about the sensitivity based on the data analysis. So in that case, what happens is the key difference between a conventional PCR technique as well as this multiplex PCR technique is that they can simultaneously detect higher number of microbial taxa, and that one key point brings about all other differences that's mentioned here. So the key difference is it can simultaneously detect higher number of samples. I hope that was clear. Is that clear? Okay. So here is the next question. Okay, two of you have answered, and the answer is right. So, genome assembly generally when we do is when we have shotguns or short sequences and we have to build back whatever the original genome that we have. So, that process is known as genome assembly. So, as rightly mentioned, genome assembly can be described as determining the order and orientation of DNA fragments to reconstruct the original sequence of the genome. The other options do not convey this meaning uh, based on what a genome assembly is. So, B best describes this. So this is a little representation of what the genome assembly is. You have all the ABC represents the genome assembly only, but you will have shotguns like this, which you arrange it to get the final genome sequence that you are analyzing initially. So that process is known as genome assembly. You can choose which of the statements is true or false. I'll just wait for 2 more minutes to see if someone else wants to answer. Okay, so the statement A is BLAST is used in the bioinformatics and molecular biology research to identify homologous sequences and infer functional and evolutionary relationships between them. So yes, BLAST, we know it's a local alignment search tool. So we can identify homologous sequences by aligning. So statement A is true, and in the case of statement B, SINA is an optimized for optimized tool for aligning large data sets, and it provides high quality alignment suitable for downstream analysis. So the downstream analysis can be phylogenetic or taxonomic studies from after the alignment. So both the statements it are true. So here is the next question. This is just a fill in the blank question. Okay, this is BLAST. So in BLAST, what we generally do is we take a nucleotide or protein sequence. What we give as input is the query, and we'll be searching it against the database, which are known as the subject sequences, and finally it will give us the output. So here we have what SINA is. It is Silva incremental aligner. So here you can use it for a multiple sequence alignment of ribosomal RNAs. Okay, so with this will be clear. So in SINA, you have this, you perform this alignment of ribosome. It's meant for ribosomal RNA, but you can perform multiple functions, which includes this downstream analysis of phylogenetic for the phylogenetic inference and taxonomic classifications. Okay, so and this is one more thing that you have to be having in mind. I think this is a repeat from the previous week session only. There are different types of blast searches: blast N, blast X, blast P, T blast X, and T blast N. So the difference will be in what input you are giving and against what database it's being searched. If you are giving a nucleotide input and it is searched against nucleotide, it is blast N. If you are giving a nucleotide input and it's being searched in a protein database, it is called as blast X. So what happens is whatever nucleotide input you are giving, it will be automatically translated into a protein, and the software itself will translate and search it in the database. In blast P, you give a protein and it's searched again, protein sequence only, and it's searched again a protein database. T blast X, what happens is you give a nucleotide sequence, it will translate it into a protein sequence, and the database also will contain only nucleotide sequence, and that will also be translated to a protein sequence, and the search will happen and you will get the results. Okay, so the use of this translating both the query and the subject is that you can identify the sequence based on their coding potential. So because we know that there is degeneracy in the code, we can have multiple codes for amino acid. So one code codes for one amino acid, but one amino acid can be coded by more than one triplet codon, right? So in that case, what happens is you have in the nucleotide you have one codon. In the database you have the another codon, but at the end of the day both the codons code for the same amino acid, then your protein sequence is going to be same only. So just to know, have some information about that, you go for this T blast X. In T blast N, what happens is blast N. So you will have this nucleotide database, but when you're going to input a protein sequence, the database will have this translated, will be translated to a protein, and against this subject your query will be searched and you will get the results. So these are different types of blasts. So based on the applications also, knowing about this is very important when you're going to perform alignment for your samples. So here is the next question about sequencing. I hope you all remember about Sanger sequencing. I explained it clearly. So here the question is about comparing high throughput sequencing methods and Sanger sequencing. Okay, so all of you have answered it. C. It is. So high throughput sequencing is generally um, sequencing many fragments at a time. So we can have a faster measurement using a high throughput sequencing. So our options are saying it gives higher throughput of sequencing, yes, obviously. Ability to sequence longer reads or higher accuracy and longer reads, and ability to sequence DNA and RNA simultaneously. So the main advantage of going for a high throughput method is having a better accuracy as well as having longer reach. So as you have mentioned it right, that's the correct answer. I've just mentioned the definition or the meaning in case someone missed out. So here is the next question regarding databases. Okay, we have a few responses here. So DASH is a publicly available database that stores DNA and RNA sequence data and associated metadata such as sequence annotations and literature references. So all the answers say it's GenBank. So let me first ask what all these four means. So what is MacVector, what is GenBank, Adobgene and Kime? Kime, yeah, we have seen already, but you can just put in the chat box about the other options. Okay, I guess. Okay, we have one response. GenBank is a collection of all genetic sequences. And as you have answered, GenBank is right. So this is about Mac Vector is a it's for MacBook, and all you can do is edit sequences, design primers, you can do alignment, you can do phylogenetic reconstruction, you can do protein analysis, etc. GenBank, again, as you have answered, it's a database which stores the DNA sequences. So one thing to note here is DDBJ and DNA are also other databases, and all these three have data exchange on daily basis, so that we don't miss out based on what database we go for our search. Uh, Chyme we have already discussed, and there is nothing called Adobe gene. So that's about the previous question. So here is the next question, easy question. Okay, so yes, proteomics is a study of proteins and their interactions with other molecules. It uses techniques including mass spectrometry, microarray, protein microarray to identify and characterize proteins and their functions. So it's right. So the study of all the proteins that are present in the sample is known as proteomics. The study of all the genes that are present in the sample is genomics. We know what bioinformatics is. We have been discussing for the past 1 hour now. Chemi-informatics is again about obtaining information from chemical data. So we go for analysis of chemicals, and then we study them for qualitative and quantitative analysis, and that is known as chemi-informatics. Okay, all of you have answered it right. So generally, if you use metagenomics to study uh, entire population of genes that is present in a given sample for soil, water, or other sample, air sample. So in that, what happens is we assemble short DNA sequences generated by sequencing into a long contiguous sequence and compare the sequences in the metagenome to these to those of the genes in that database, and then we try to identify and annotate the genes. So by this what we do is we get to know the functional potential of the microbial community that is present. So generally this is the flow. So what we have is any sample, a soil or water sample, and we don't know what all the microorganisms are present in it. We just take the sample, give it for sequencing. So once we get the sequences, we just go for a database search and see where it pairs, and based on that we try to annotate the functions also. So based on the functions we'll know what gene codes for what protein. So based on the availability of or the expression of these proteins, we know when this protein is expressed what will be the type of bacteria or the function of that bacteria that's going to be present in that given sample. So this is known as a metagenomic analysis. Metagenomic analysis can be done for any thing or any surface, just to study about the entire microbes that are present. One more easy question, not too much to read. Okay, we have A and C in the answer, but here it's A. All others are techniques, but in the samples, what you have is you're going to collect some particular targeted thing and then do the analysis of it and check. So basically, 16S rRNA, it is a ribosomal RNA. Okay, the S here stands for Swedberg units. We have discussed when we were studying about the organelles that were present. So this helps in the study of phylogeny and taxonomy because it's the most common housekeeping genetic marker. Okay, and the other reasons why we use 16S rRNA is it's present in almost all the bacteria, and the function of the 16S rRNA has not changed much over time. So you will have the consistency while doing this study, suggesting that random sequence, it's very conserved with evolution, and it's large enough for to study for the informatics purpose. To rely on the data, it's large enough. It's around 1500 base pairs long. One more advantage is you would just go for a culture free method in this. As you told, you go for sequencing methods or culture free methods. So for that, you use the 16S rRNA. So this is just the open-ended question. Name few DNA databases. GenBank, okay. Are you aware of anything else? Okay, NCBI. Okay, just for your information, you have all these DNA databases. You have GenBank, which is the most widely used, and it's from NCBI. So you get GenBank from NCBI, and you have DDBJ, DNA database of Japan. As we saw, we also have that ENA. We have this TIGR. Um, we have East for East genome, and we have one more called this microbes. So all these are few DNA databases. Uh, Mainly, at least you should be knowing one or two databases. Blast and Kime are basically not databases as such. You also use them as tools for analysis. So, but what I have asked was generally DNA databases. Okay. So yes, again now, name few protein databases. You should be clear about what is a software, what is a tool, what is a database, what functions they do, and what information we get from each of them. So, a clarity about this is very important to fetch the right information from these from these databases, tools, or softwares that are available. If you want to perform a certain function, you should know what tool to use. If you want to search for something, you should know what you have to be using. If you have to do a uh, sequence alignment, or if you need to find out the phylogenetic relationship, so you have different tools for all of these. When you know what your purpose is, you have also you should also be figured out what is the tool that you should be using for that purpose. Right, and this is a reminder to anyone who is new to all of this, who has very less exposure to bioinformatics before. The reason why I'm asking all these questions is to make you understand what's the difference and what different things you have to look up in the case of bioinformatics. It's not just few lectures or sessions you sit and you learn bioinformatics. That's not how things work. You can just go and search for yourself. I'm just giving you an idea of what kind of stuff is present for you to go and search and study. Okay, so now you know for what you use a database or for what you use other tools. In database also you will have this DNA database, protein database, etc., and they can perform different functions also. You can just go back and check and have a clear idea of what each of it is. So we have Swissprot, which has very high level of annotation. So you have a very refined data about proteins there. You have protein information resource, which is powered by this UniProt org only, whereas the Swissprot is from Expesi. And again, this is a very good database for protein searches. You have something called translated databases. So here you have this translation data also in them in those databases, which includes this Tremble, Genpect, and PDB. And PDB is very commonly used protein databases. It's a protein data bank. So when I think whenever you search for a protein, or when you want to find the sequence, when you want to find the structure, anything, you will most probably use PDB. And you would have heard people telling about PDB ID of a particular protein. So there is unique ID for all the proteins that are submitted to PDB. So with that unique identification thing, you can just go for it and check. It will also have information from what study was performed, or if like NMR or X-ray crystallography, what kind of analysis was performed to elucidate information about that protein, and from what organism that protein was isolated. All such information you can find in PDB, and it's one of the most commonly used protein databases for many purposes. So the last question is about scoring matrices. You can just answer what are scoring matrices. Okay. Uh, any of you have any idea about what a scoring matrix is? Okay. Basically, we perform this sequence alignment and stuff. So at the end of which you will get some score. So that score tells about the reliability of that particular result you have obtained. So you will have individual scores for see you're going to enter in what. Okay, one more question. Okay, I'll answer that after explaining this question. So when you're going to perform a sequence alignment, what happens is um, you're going to enter multiple base pairs as input for that alignment, and after that you will get the result and you will get a score, right? So in that score, what happens is it's based on the number of mismatch that has happened, and there is individual score for all of this, and finally you will get the output score. So this cannot be performed individually when you are going to have this multiple base pairs. I mean, you will have kilo base pairs of data or something like that. So what happens is you go for this scoring matrices, where we use this matrix to tell what is the final score or the reliability of the results that we have gotten. So with that score, we can know how to proceed with further data analysis. So after the alignment, you will get what is the alignment and what it means. But after that, how to proceed. So when you're going to analyze the data, in what should be the next step? So to determine that, you will have the score as one of the criteria for validation. So this scoring is done using a few matrices, which include PAM matrix and Blossom matrix. So PAM is the percent accepted mutation. So as I told you, you have scores for every nucleotide match and every mismatch. So based on this, what is the accepted mutation that can happen in the sequence? And in case of Blossom, you have this like when you're going to do for the proteins, you have this blocks amino acid substitution matrix. So they have their own algorithm based on which they work. If you are interested in knowing, and if you are, if it's required for what you'll be working on, you can just go back and read. There are multiple resources available to study about Pam and Blossom. You have even standard textbooks that explain this, and you can also go and read back to the literatures that had this matrix algorithms published first, so you will have a better idea of how things work. I think all of you would have heard about local alignment. So local, we have two things called local alignment and global alignment. So in local alignment, what we do is we try to match local areas in the gene. In global alignment, what we see is the whole matching, whole alignment of the entire set of input that we have given. That's the difference between local and global alignment. So we have few algorithms based on which this alignments all work. So here we have this fast alignment algorithm BLAST, and then the main thing around which this local alignment works is the Smith-Waterman algorithm. And for global, you will have something called a Needleman-Wunsch algorithm. And here you have the Smith-Waterman algorithm. All these is not to intimidate you, but just to tell you that all these aspects of bioinformatics is available. So whatever is the requirement for your course or for your work, for whatever purpose you are studying this, you can go back and search and use all of it. Okay, how sequencing depth affect the accuracy of the sequencing run? So sequencing depth, okay, I'll go back to that question. So here what we have is sequencing depth means the average number of times a given position in the genome is sequenced. Okay, so if I'm going to sequence it for one time, and if I'm going, if that happens for some 10 times or 100 times or 1000 times, I mean that every with every increment, the sequencing depth is going to increase. Okay, so here what I'm asking is how it affects the accuracy of this sequencing run. So what we mean here is, if I'm going to repeat the process for some, say, 10 times, 100 times, 1,000 times for a particular position of in that genome, the accuracy will increase, because first time I'll get something, after 10 times I'll get some reading. With that, what I can match is, okay, if A is being repeated many times, even if I do the sequencing in, let's take some 12th position in that genome, I'm performing it multiple time, and maybe out of 10 times what I got is, okay, first time I got some T in that position. So that was first thing. When I'm doing the same thing for 10 times, I get some 2 T's and rest 8 as A. So what happens is that accuracy is increasing here, right? So if I'm going to get multiple times of A in that base, that means that 2T that I got was because of some error. Like it was a random chance. It was not the actual thing that had to be present there. So with sequencing depth, your accuracy of the read will increase. When you perform it, when there is random chance of happening, when you perform the experiment, you can confirm that no, that was not random, but that was something meant to be happened. This is basic for any experiment you do. Okay. Here what we are trying to tell is, but this accuracy is not going to be infinitely keep on increasing when you're going to increase sequencing depth, because at one point you will know what is present in which position. So that this thing will attain a plateau. Your accuracy keeps on increasing up to one point, and here you know for sure that this is what is going to happen. So even if you are going to perform uh, the same experiment again and again and again, you're going to get only that value or that reading. So at that point, what you call is it goes to a plateau. Your accuracy no more increases. When you finally get all things right, your maybe to tell it very simply, I can tell that, see, I give you an exam of 100 marks. So initially you score some 80 marks. Maybe next time I give you the same question paper, you score 85, you score 90, 95, and finally when you score 100, you will reach a plateau. After that, even if I give you the question paper for some 10 more times, the maximum that you can score in that is 100 only. So your accuracy will keep on increasing under some time, and after that it will reach a plateau. So that is what is the meaning of this question. I hope it is clear now. Okay. So yes, that's it for today. If you have any doubts, you can ask. Thank you for joining in for this session. I hope the session was informative. Bioinformatics has a lot to ponder in and search, and you do it as and when required for maybe your research or for some other thing that you will be requiring, or for a particular teaching purpose or something. So you can always have, you also have many resources online because it's informatics again. You have many online resources, so as and when required you can refer to them. You can also refer to some standard textbooks. um, if you are interested in knowing about the backend operations of any software or developing them in that case. So with that, like, I think we can conclude the session today here. Thank you all for joining in. Hoping to see you tomorrow for the last session, which will be the revision session. So I'll be discussing topics from all the 12 weeks, a quick overview of all the 12-week course content. If you have any specific doubts, please remember to post it in the discussion forum so that I don't miss out on discussing that tomorrow. You can also come and ask in the class, but if you post in the discussion forum, I'll just be prepared and I'll just add it in my presentation as well. Thank you all for joining in.